Hi, Assalamualaikum, Madam Munira. My name is NADIRA binti Muhammad Nizam from DBT 3. My metric number is 23 GPT 20F 2017. Today, I would like to present a virtual experiment simulation of Sds Page. So first of all, we need to know the theory of Sds Page. Sds Page are sodium dodecline sulfate polychloramide gel electrophoresis is a technique used to separate of protein based on their molecular weight. This is a technique widely used in forensic, genetic, biotechnology, and molecular biology to separate the protein molecule based on their electrophoretic mobility. So next, we're going to talk about the principle of Sds Page. The principle of Sds Page states that a charged molecule migrates to the electrode with the opposite sign when the place is an electric field. The separation of the charged molecule depends upon the relative mobility of charged species. The smaller molecule migrates faster due to the less resistance during electrophoresis. The structure and the charge of the protein also influence the rate of the migration. Sodium dodecline sulfate and polychloramide eliminate the influence of structure and charge of the proteins, and the proteins are spread based on the length of the polypeptide chain. Next is the role of Sds in Sds Page. Sds is a detergent present in the Sds Page sample buffer. Sds, along with some reducing agents, function to break the disulfide bonds of protein and distribute the hierarchy structure of proteins. So now, let's begin our experiment. Firstly, we need to increase the heat block temperature to 95 Celsius. Next, set the micropipette to 30 microliter and attach a tip. Draw up the wild-type cell culture. Close the wild-type cell culture and dispose the wild-type cell culture into the wild-type cell tube. Repeat the procedure to transfer 30 microliter of the pancreas cell culture. Then, repeat the procedure to transfer 30 microliter of the added for cell culture. Last but not least, repeat the procedure to transfer 30 microliter of the added 7 cell culture. Next, set a micropipette to 10 microliter and attach a tip. Next, draw up 10 microliter of 4 times SDS sample buffer. Don't forget to close the buffer sample. Next, dispose the 4 times SDS sample buffer into the wild-type cell tube. I'm going to repeat the same procedure for the pancreas cell tubes. Edit 4 tubes and edit 7 tubes. Four times SDS sample buffer solution is used to prepare the protein sample for gel electrophoresis. Move all of the samples into the heat blocks. Function of the heat block is used to incubate sample at a constant temperature over a long period of time. We're going to heat the tubes about 10 minutes. After done, remove all of the tubes from the heat block. Move all of the sample into the microcentrifuge. Don't forget to balance the tube in the microcentrifuge. This is because to provide enough weight to balance out the other microtubes. After that, place the polycalamide gel into the electrophoresis box. Fill the gel cassette with one-time running buffer. After that, we can remove the comb. Fill the outer gel electrophoresis tank with one-time running buffer. Set a micropipette to 20 microliter and attach a tip. Draw up the protein ladder. And move the micropipette to the first lane of the protein gel. After that, we're gonna repeat the same procedure to fill the other lane using the NADIRA samples. Okay, next, connect the red and black cables to the power supply and the lid of the gel electrophoresis box. Sure to connect the cables with the right place, which is red with red and black with black. Now we can turn on the power supply. Set the voltage to 150 volt while the timer is 90 minutes. We can start the power supply. Wait until the time has elapsed and turn off the power switch. Then, unplug the electrodes and inspect the gel. And finally, our experiment is done. Thank you, Madam Munira.